Food first enters the oral cavity, then gets mechanically digested by the teeth. Then amylase is released from the salivary glands, which initiate the digestion of starches, which get converted into maltose or a smaller carbohydrate. The canine tear the food and the molars are used for against taste buds. 30% of taste is oral and 70% of taste is what you smell. The salivary glands contain amylase, which initiate the digestion of starch. The salivary glands contain mucin, which keeps the bolus together. There's also exocrine glands, which use ducts to move secretions inside of the bloodstream. Behind the mouth is the pharynx, where the nose and the mouth meet. That is where the tongue pushes the bolus to where it is swallowed, passing the epiglottis and going through the esophagus. The esophagus is roughly 25 to 30 centimeters long. It is made of smooth muscle which secretes mucin. Food is moved down the esophagus through peristalsis, which involves contractions of the smooth muscle until the bolus reaches the stomach. While food is in the esophagus, no digestion occurs, and this process is involuntary, and the food travels down the esophagus. Once the bolus has passed the diaphragm, it enters the stomach, where mechanical and chemical digestion occurs. Instead of the stomach, the bolus meets the cardiac sphincter, which are circular materials that regulate the passage of material. The cardiac sphincter controls the opening of the stomach to the esophagus. The pyloric sphincter regulates the movement of foods and stomach acids entering the small intestine. Within the stomach, the bolus causes the stomach to expand, which happens through a wall of millions of tiny gastric glands called rugae. The gastric glands release gastric juice per meal to help with digestion. Muscle contractions mix the food and gastric juices. The gastric glands have multiple cells to protect the stomach from bacteria as well as kill bacteria. The mucosal cells produce protective coating. The partial cells secrete hydrochloric acid to kill bacteria. And the peptic cells secrete pepsinogen, which aids in digestion once turned into its active form. Before entering the small intestine, the food turns into a semi-liquid form called chyme, which is what food is called before entering the small intestine. Chyme tends to have an extremely low pH, making it highly acidic.
After food is chemically and mechanically digested in the stomach, it enters the small intestine. The small intestine is split into three different parts. The duodenum, which is represented by the orange color in the drawing, where majority of digestion takes place and is the first 25 to 30 centimeters of the small intestine. The jejunum, which is represented by the red color in the drawing, is the second part of the small intestine that is responsible for absorption and is 2.5 meters long. The ileum, which is represented by the blue in the drawing, is the last section of absorption in the small intestine, which is about 4 meters long. Food enters the small intestine from the stomach through the pyloric sphincter. The pyloric sphincter regulates the movement of foods and stomach acids entering the small intestine. When food eventually does leave the stomach and enters the small intestine, it becomes chyme, which is the food in a paste form which is highly acidic. This acidity/low pH triggers prosecretin in the small intestine and is activated into secretin by the hydrochloric acid. The secretin then enters the bloodstream and travels to the pancreas. After the secretin reaches the pancreas then releases the bicarbonate ions, raising the pH and creating a more basic environment. This basic environment inactivates the pepsin secreted by the gastric glands. The pH is raised in the small intestine to prevent ulcers. The pancreas also secretes trypsinogen into the small intestine, which is activated into trypsin by enterokinase. Trypsin helps digest the proteins that were partially digested by the pepsin in the stomach. The trypsin breaks down medium-chain peptides into shorter-chain peptides. The small intestine then secretes erepsins, which finish off protein digestion in the small intestine by breaking down the shorter peptides into amino acids. Carbohydrate digestion continues in the small intestine after being partially digested in the oral cavity by salivary amylase, and continues by the pancreas secreting more amylase, which breaks down the simpler carbohydrates into disaccharides. The small intestine then secretes disaccharidase, which break down the disaccharides into monosaccharides, which finish off carbohydrate digestion. Lipid digestion takes place when lipases are secreted by the pancreas. When lipids are detected in the duodenum, it triggers a release of the hormone cholecystokinin, CCK, which trigger the release of bile salts. Bile is produced in the liver and stored in the gallbladder. The liver not only produces bile but also stores glycogen, vitamin A, vitamin B12, and vitamin D, and filters the blood. The bile salts are then transported from the gallbladder into the duodenum through the common bile duct. Bile salts assist the lipases in lipid digestion by emulsifying the fats, increasing surface area of the lipids, which speeds up chemical digestion because the lipase is exposed to more of the lipid substrate. Throughout the small intestine, all this effective absorption would only be possible through villi, which increase surface area by 10 times. Villi also have infoldings of their cell membranes called microvilli, which even further increase the surface area for more effective absorption. The capillary network is located in the small intestine wall, which are tiny blood vessels where carbohydrates and amino acids are absorbed. These are intertwined with lymph vessels called lacteals, where fats are absorbed. The time taken for the digestion process to pass through the small intestine is around the 5th hour in the entire digestion process.
The large intestine's main purpose is for water reabsorption and for the waste removal, as chemical digestion is complete by the time it reaches the large intestine. First, the digested food moves into the ascending colon, then the transverse colon, then the descending colon, and after that sigmoid colon. The colon is the largest portion of the large intestine, containing intestinal bacteria, and stores the waste long enough so water can be reabsorbed. The colon also contains mucus to help move digested foods towards the anus. The bacteria in the colon use the water products to synthesize vitamins B and K. Then the water is transported into the rectum, which is a temporary waste storage area until the waste is removed through the anus. The anus is the opening where the solid waste is removed from the body. Bowel movement in the process are crucial, as they ensure that potentially toxic waste are removed from the body. Bowel movements are caused by cellulose, which is from the plant material that we eat, and provide bulk in the colon to cause the bowel movement. Also, bowel movements are caused by high fiber foods as well. The cecum is a pouch which connects the small intestine to the large intestine and is the first part of the large intestine. The cecum has no apparent functions in humans. Also, the appendix is a vesical organ, meaning it has no function either. The appendix is only noticed when it gets infected, which causes severe abdominal pain and can be dangerous to the body, as it sends out microbes into the body cavity.